Hi, my name is Angelica Arevalo, and I'm from New York in the United States. The title of my research is Chemotherapy Mediates JAG1 Macrophages and Invasive Cancer Cells. Paclitaxel is a commonly used chemotherapeutic drug agent to treat breast cancer patients. However, studies have found that over time, 90% of these patients develop chemotherapy resistance, which in turn may lead to increased metastatic risk. This process is known as chemotherapy-induced metastasis, and it poses a major clinical obstacle today due to lack of understanding. And thus, I sought to further elucidate the underlying metrics behind this process. Studies have found that paclitaxel paradoxically induces an influx of JAG1 TMM macrophages. These macrophages are known to play a very crucial role in the assembly and function of tumor microenvironment of metastasis, or for short, TMM sites. And thus, this alternate reaction can lead to a plethora of negative events that could snowball. These events include TMM density increase, MENA-invasive upregulation, tumor metastasis, and eventually paclitaxel resistance. However, this is not fully understood how. In a study done by Reddick et al. In 2005, it was found that the co-expression of the JAG1 Notch ligand and the Notch1 signaling receptor is associated with poor overall survival in breast cancer patients. This indicates that the Notch signaling pathway may be at play here. In another study done by Andrew et al. In 2016, it was found that the overexpression of the JAG1 Notch ligand promoted a shorter distant metastasis-free survival. In another study done by Cabrera et al. In 2016, it was found that Notch1 signaling mediated the uptake of MENA-invasive tumor cells in the tumor microenvironment. These tumor cells are known to exhibit a migratory phenotype and are likely to metastasize. More interestingly, in another study done by Cabrera et al. In 2018, it was found that the JAG1 expression promoted the epithelial to mesenchymal cell transition in breast cancer tumor cells. This biological process is known to allow breast cancer cells to acquire their migratory phenotype, and thus it proposes that a TMM macrophage and TMM tumor cell do interact with one another. However, the Notch signaling pathway has yet to be characterized within this cascade. In my study, I proposed that the MENA-invasive tumor cell and the macrophage, which both play very crucial roles in the function and assembly of TMM sites, interact with one another upon the binding of the JAG1 Notch ligand and the Notch1 receptor. However, I wanted to use DAPT, a Y secretase inhibitor, to inhibit the Notch signaling pathway and to determine if it could have any effect on paclitaxel resistance, as this is an approach that has never been done before. And similarly, I wanted to use conjugate liposomes as a macrophage depleting agent to determine if it could have any effect on the population of MENA-invasive tumor cells in the tumor microenvironment. The purpose of my study was threefold. I first wanted to assess the behavior of macrophage cells in breast cancer tumors. Second, to elucidate which Notch ligand contributes to MENA-invasive expression. And lastly, to elucidate the efficacy of Notch inhibition and conjugate liposomes on breast cancer tumors. I hypothesized that paclitaxel will increase both JAG1 macrophage density and MENA-invasive tumor cells. Second, that DAPT may be able to mitigate this influx of JAG1 macrophages, and similarly, that conjugate liposomes may downregulate the overexpression of MENA-invasive tumor cells. In my study, I chose to utilize the female MMTV PYMT mice model for its accuracy in portraying tumor metastasis and progression in breast cancer tumors. All of the mice were randomly selected to be part of two different experiments. In the first experiment, I was analyzing JAG1 macrophage density, and in the second experiment, I analyzed the expression of MENA-invasive tumor cells. From here, all of the mice were randomly assigned to different treatment groups, all of which were compared to a control. I'd like to emphasize the development of my fourth treatment group, in which I call the combination group. The purpose of the combination group was to determine if DAPT or conjugate liposomes may have any effect on paclitaxel resistance. At week 9, all of the mice were put to undergo a treatment timeline for 2 weeks according to their treatment groups. And at week 11, all of the mice were sacrificed, and tumors that were 5 millimeters in size were extracted from the mice. All the tumor tissue was prepared for immunofluorescence analysis, and after immunofluorescence analysis was completed, all the slide images were uploaded on a panoramic 250 whole-scale digital 2.0 scale scanner. To begin image analysis, I first quantified 10 regions of interest from the tumor tissue at 40x magnification. And then within each region of interest, I calculated the number of positive macrophages. And furthermore, within this population of macrophages, I elucidated how many of them expressed the JAG1 Notch ligand. All of the images were analyzed on image J, and the data was organized on Excel spreadsheets. I also conducted statistical analysis using GraphPad Prism 6, in which a p-value of less than 0.05 was considered statistically significant. Now, what did I find? I first hypothesized that paclitaxel will promote an increase of JAG1 plus macrophages, and my findings support this. As you can see here on the graph on your left, there was a significant uptake of these macrophages compared to a control. My findings indicate that paclitaxel recruits macrophages into the tumor microenvironment via Notch signaling. And thus, I wanted to use DAPT to inhibit the Notch signaling pathway and determine whether it would have any effect on the population of JAG1 macrophages. And I found that there was a significant decrease of this macrophages upon the administration of DAPT. My findings support that not only that DAPT can inhibit the Notch signaling pathway, but it revealed that paclitaxel and DAPT play a very distinct role within this cascade. I also wanted to determine if a similar finding can be translated into the combination treatment group. And my findings support that paclitaxel and DAPT showed less macrophages compared to the paclitaxel treated group. My findings also reveal that DAPT may mitigate the effects of paclitaxel chemotherapy resistance. In the second experiment, in which I was analyzing the expression of MENA-invasive tumor cells, as hypothesized, I found that paclitaxel promoted an influx of these tumor cells. Therefore, I wanted to determine if the use of conjugate liposomes can mitigate this effect. And it did. As my findings show, there was a significant decrease of these tumor cells through the use of conjugate liposomes. However, the main purpose of my study was to also determine if this finding or idea could be translated into the combination treatment group. And I found that there was a significant decrease of MENA-invasive tumor cells in the tumor microenvironment in the combination group compared to the paclitaxel treated group. Similar results were also found in an HT17 xenograft model. And my findings support that in both models, paclitaxel promotes the upregulation of MENA-invasive expressing tumor cells. However, the administration of conjugate liposomes can mitigate this effect. A review of major findings. First, I found that macrophage tumor cell interaction mediates the Notch signaling pathway. And second, I found that paclitaxel utilizes the Notch signaling pathway to induce pro-metastatic macrophage cell behavior. This was exhibited through the influx of JAG1 macrophages and MENA-invasive tumor cells upon the administration of paclitaxel. However, I also found that both DAPT and conjugate liposomes respectively undermine the side effects of paclitaxel administration in mammary tumors. A novel aspect of my study was that I analyzed all of these images blindly, and this was done purposefully to reduce bias. However, there were some limitations in that I did not calculate TMEM score, and I only accounted for one Notch signaling pair. However, future research can address this by conducting a similar study and looking at different Notch pairs, calculating TMEM score, conducting invasive assays, but more importantly, comparing the efficacy of Notch inhibition for more chemotherapy drugs. Some novel applications of my research is that I propose the Notch signaling pathway as a biomarker of tumor metastasis and paclitaxel resistance. However, it is important to note that both of these processes are part of a greater cascade, and it can improve the better understanding of the metastatic cascade, as it is a complex biological process that researchers have yet to fully understand. My findings can also translate into the development of more novel and efficient chemotherapy drugs to treat worsening or relapsing breast cancer patients. In conclusion, in my study, I inhibited the Notch signaling pathway in a murine model to better understand chemotherapy resistance and breast cancer. I found that DAPT reduced JAG1 macrophage density by inhibiting the Notch signaling pathway. And I also found that conjugate liposomes suppress the expression of MENA-invasive tumor cells. My findings indicate that the Notch signaling pathway plays a very crucial role in paclitaxel resistance. I would like to thank my mentors, Dr. Akhte and Dr. Karagiannis, for all of their help, and my science research teachers for their guidance and unwavering support throughout these pressing times. I'd also like to thank my friends, family, and peers for all of their feedback.